So everything in this PowerPoint, we'll look at what are characteristics of viruses. A lot of the PowerPoint is how do viruses replicate. We'll also talk about how can viruses cause cancer, because they can. How and why do we want to culture or grow viruses? And then that kind of, are viruses alive? Technically no. But we talk about them as they're living organisms. And then again, we wrap up at the very, very end. What are prions and why do we care about prion diseases? So what makes a virus a virus? A lot of it's based on its size, but that's not the only thing. But some characteristics of viruses. One, they are extremely tiny. They're not cells. So I've got listed up, they're acellular. They're not a prokaryotic cell or a eukaryotic cell. They don't have cell membranes. They don't have organelles. They don't have cytosol. Like, they are not a cell. They infect cells, but they themselves are not cellular. Genetics, they can contain either DNA or RNA, and that's how we'll discuss them. So on Wednesday, we'll talk about the top DNA-containing viruses that cause disease. And then next week are the RNA viruses that cause disease, and there are more RNA virus diseases than DNA. Even though they're not a cell, they cause tons of different types of infections and diseases. Whether they cause diseases in humans, which we care mostly about in this class, but they cause diseases in plants, they cause diseases in other animals, they can cause diseases even in bacteria. They don't do any kind of metabolism, so you don't feed them and they break down food. They instead rely on their host cells. They can't respond to their environment, so they can't swim away from something. They can't react to an environment. They can reproduce, but not on their own. They need their host cell for that. And we find viruses in one of two states, either extracellular or intracellular. Extracellular just means it's not inside of a cell yet, and how our immune system reacts is differently. Intracellular means it's now inside of our cells, and our immune system reacts differently. If you remember back when we did the immune system, I drew up the two branches, and I'm like, our B cells are targeting extracellular things, and our T cells target intracellular things. And again, those cells can recognize whether viruses have gotten into a cell yet or not, and target them differently. In an extracellular state, so this means a virus has not gotten into whatever cell it wants to get into, we call them virions. It's another name for them. And they consist of two things. They consist of a capsid as well as nucleic acid. And nucleic acid, that's the NA of either DNA or RNA. So it's got an outer protective capsid, and then it's got the genetic material. Structure-wise, we smush the two words together, because why not? And we call it a nucleocapsid. Again, it's got this outer capsid, kind of peachy colored structure, and then it's got the genetic material in it. Some have a phospholipid envelope, but not all. So I have phospholipid envelope, but not all viruses have it. And when we start getting into viruses on Wednesday, you'll see some of them listed. Are they enveloped or not? Do they have that outer phospholipid envelope, that membrane, or don't they? If they have it, we call them envelope viruses. If they don't, we call them naked viruses. It just means they don't have an outer envelope. And because they're not inside our cells yet, the humoral branch of our immune system is recognizing and targeting them. So your B cells. Once they get inside of our cells, which is their ultimate goal, once they get inside the cells, they get rid of the capsid. They don't need it. The biggest part, because again, the only thing a virus consists of is a capsid and DNA or RNA. Once they get rid of the capsid, what's left is the genetic material, and it gets inside of the cells. So the only thing that's getting inside of the cells that it cares about is that nucleic acid. Again, the NA of either DNA or RNA. Once it's now inside of the cells, Now it's our cell-mediated immune response branch, or our T cells, that have to be activated to target them. Now, for the genetic material of viruses, there is a lot more variety on what kind of genetic material a virus can have. Again, we're like, well, can it have DNA or RNA? Yeah. But there's a variety just in that alone. So based on what kind of genetic material it has, we can use that base to categorize them and classify them. Again, Wednesday's lecture is a whole chapter on DNA viruses. And then next Monday and next Wednesday, which is recorded, is all RNA viruses. Because even though it can have DNA or RNA, and you're like, that's not a lot of choices, there's still some differences. Because you could have double-stranded DNA, or you can have single-stranded DNA. You can have double-stranded RNA, or you can have single-stranded RNA. But there's even a difference in the single-stranded RNA. I'm going to go back to central dogma. DNA makes messenger RNA, makes protein, which I don't care about the protein, but I'm going to leave it up because I guess I will talk about it. Generally, DNA is double-stranded. Generally, messenger RNA, or RNA in itself, is single-stranded. When we go from DNA to RNA, when we go from double-stranded DNA, 2 strands, to a single strand of RNA, the question becomes, well, when we copy the DNA to make your single strand, did it copy that strand, the top one? Or did it copy the bottom one? There are 2 strands in double-stranded DNA. Depending on which strand is copied, depends on which single strand of RNA you have. So there's even a variety in the single-stranded RNA. We call them either positive or negative. Just know, because you're going to see that coming up. Because there are 2 strands of DNA. So there really are 2 options depending on which strand gets copied. They're opposite of each other. So that's why we call one positive, one negative, because they're opposite. So if there was an A on here, there would be a U on that one, and vice versa. The genetic material can be linear and segmented, which means all the genetic material, we have it in lines, but it's found in little segments. When we get to it, the influenza virus has linear segmented, which means all of its material, all of its thing, it's in lines, but it's segmented. It's not just one long strand of RNA. Sometimes their genetic material can be small and single and circular. Example, this would be a viral genome where we have just a single circular genome, where it's one continuous strand. But either way, depending on whatever kind of genetic material they have a lot less genetic material than any other living organism because they're not technically alive. So all of these squiggles up here is the viral genome. This is the DNA found in E. coli, and you can't even see all of it because some of it goes off this picture. This is an actual electron microscope picture of the DNA of E. coli, which is still a tiny little organism, compared to the DNA of a virus. So they have a lot less information. Again, it doesn't mean they don't cause serious issues, but the little information they have, the little bit of DNA or RNA they have, is a lot less. Now, the viral hosts, what viruses attack is very specific. So they infect very specific cells. It's usually because the cells they infect have some unique protein on the outside, and that's what the virus recognizes, and that's what it can attach to. If the cell didn't have that unique structure, virus couldn't attach, virus couldn't infect. Now, some viruses are not as specific, and that's usually just because there are a lot of cells that might have that one little protein on the outside. So the viruses, anything that has that one protein, the virus is going to attack all of them. Others are more specific. This is just showing, this is a very specific virus that's infecting this very specific plant called the tobacco. I was going to say it's just the tobacco plant, but it's a virus called the tobacco mosaic virus that only attaches to and attacks that very specific plant. HIV is a virus that affects very specific cells in humans. We're talking your T helper white blood cells. So it's not affecting your other cytotoxic T cells or any other cells in our body. It's specifically the T helper cells. And then there are bacteria that are there are viruses that are very specific for attacking bacteria. So if you're like, well, bacteria are bad and viruses are bad, they affect each other? Absolutely. There are lots of viruses that infect bacteria. Viruses that infect bacteria are called bacteriophages. And that confuses a lot of students because it's got the word bacteria in it, so they think it's a bacteria, but it's not. It's a virus that infects bacteria. So a lot of times we take the word bacteria out and we just call them phage viruses, but a phage virus means it's a virus that is infecting a bacteria. So here's, I don't know if it's an E. coli, it's some kind of bacilli shape. Tons and tons of little viruses that are all attaching and attacking that one little bacteria. Size of viruses, they are tiny. Before we had electron microscopes, we didn't know they existed. I mean, we called them germs, but we couldn't see them, we couldn't study them, we didn't know a lot about them. So this is just looking size comparison, a red blood cell, eukaryotic cell, compared to an E. coli. Super tiny, but if you magnify it enough, we can see it. If we zoom in on that E. coli, maybe you're like, oh, there are some tiny little things next to it. If we zoom in on that E. coli, we can start to see some of the size differences of viruses compared to bacteria. And you can even see the variety just in sizes. There are some viruses that are quite large. The smallpox virus is one of the largest viruses, which if you had a good enough microscope, maybe you'd see something that looked like a dust speck next to E. coli, but you wouldn't see anything more than that. Down to some of the tiniest of the viruses, which is the polio virus. So there's a big difference in sizes, but either way, they are still extremely tiny compared to bacteria. Again, they're not a full cell. They don't need all that stuff. They're just genetic material and a capsid, and some have a bonus envelope. So that capsid, and why we care. So that capsid that it has in its extracellular state, it's really there for protection and to recognize the cells that needs to attach itself to. It is a protein coat, so it's made up of proteins. And all the tiny little circles that make up the capsid are called capsomeres. It's like the little tiny fragments. So each one of these little circles are the capsomeres that make up the entire capsid. Ignore page numbers. I couldn't guarantee that that's accurate because they change. And again, that capsid, it's that outer protective protein coat. It's there for protection. It's got to protect that genetic material that's inside of it. It's there to recognize the cell that it wants to attach to. So it's looking for a specific receptor on a cell to attach to. And it can give it some really cool shapes. It's kind of like extra. I don't know if it benefits the virus, but it helps us identify them. Because some capsids, I'm going to go back. Some capsids kind of look like these icosahedral shapes. A lot of viruses do. Get my clicker. Some look like they should be landing on the moon more than anything else. And a lot of the bacteriophages, so the viruses that infect bacteria, look like this. There aren't any viruses that infect humans that look like something that should be landing on the moon. They are cool looking though. Some capsids are super long cylindrical, and there's one that we'll talk about too that the capsid looks bullet shaped. So the capsids give it shape, which again can help us identify it. That's for our benefit. The benefit of the capsid for the virus protects the genetic material, recognize the cells it needs to attach to, and then attaches to them. Now, as I said before, not all viruses have an envelope. If they do, we call them enveloped. Where they get the envelope from is they steal it from their host cell when they get released. And you'll see that in some of my pictures when we talk about replication. As the virus leaves the cell, it steals the cell's outer membrane, and that becomes the envelope. Because it's stealing it from its host cell, the envelope is a phospholipid bilayer, because that's what our cell membranes are. And then it does have little glycoproteins, little carbohydrate protein spikes that are very specific for it. And it's these glycoprotein spikes that are then recognizing other cells, attaching to them and hijacking them. Now, you would think that if you're a virus, so it's got its genetic material, it's got a capsid, and it has an envelope, you would actually think that by having this extra outer envelope would be beneficial to the virus, that it would allow it to survive longer, but it doesn't. Viruses that have evolved to steal and have these envelopes actually don't survive as long. These envelopes are very susceptible to drying out. They're very susceptible to alcohol, which means they're a lot easier to kill. And so these generally envelope viruses usually don't cause as extreme diseases because they're easier to get rid of, or as many diseases. So even though you're like, that's an extra outer layer of protection, that's easy to get rid of. And then it needs that extra outer layer of protection, but we get rid of it. So a lot of envelope viruses are easier to kill. This is just showing another envelope virus. Again, the capsid shape it's still got an outer membrane, but that capsid shape can still change on the inside. It still has little glycoproteins specific to it. Does anyone know what this looks like or recognize this particular virus? I was going to say maybe we're beyond it already. It's in the group of coronaviruses. Specifically, this one causes SARS, but it looks just like COVID-19. It's all in the same grouping, so under electron microscope, they look the same. And again, we can actually see these little glycoprotein spikes that stick outside of that envelope. Now, viral replication specifically for viruses that attack bacteria. So we're first going to talk about viruses that attack bacteria, and then I'll talk about, because they're very similar steps, how they're a little bit different for viruses that attack and attach to humans. Now again, all viruses are dependent on host cells. They cannot reproduce on their own. Viruses that infect bacteria go through what's known as lytic replication. It has that word lysis in it, and the word lysis means to break apart. Even in our lab, we look at hemolysis. That's breaking apart of red blood cells. Lysis means to break apart. So lytic replication means in the end, the host cell is going to die. It's going to break apart when it releases viruses. Now, it goes through 5 stages, and the book has started to rename stages, so that's why I've got 2 of them have 2 names on there. I was like, well, that's new. They used to, I don't want that marker. They used to call it attachment, which I like better than the new renaming called adsorption, because to me it sounds like it's going into the cell at that point, like absorption, but it's not. It just means attaching. And I'm going to talk about these in my pictures. Then it has to get into the cell. So it used to be called entry. Now it's called penetration. Entry was shorter. I liked it. Synthesis. The word synthesize means to make something. So they named it replication. And we're making stuff. And then they left the last 2. Assembly and release. I'm writing these up because I'm going to add some more stuff to it shortly. Again, it has to attach, adsorb, to a cell. It's recognizing some receptor on that cell so that it knows that this is the cell that it can infect. So it's got that attachment or adsorption. The entry or penetration, the only part of the virus that needs to get in the cell is the genetic material. The rest of the capsid can stay out. I mean, sometimes it gets in there, but then it gets rid of it right away. The only thing we care about is getting the DNA or RNA inside the cell. That's the penetration or entry. The synthesis or replication is, let's make the parts. There are two things that a virus is made up of. It's made up of the capsid, and the capsid is protein. So we got to make some proteins, and nucleic acid. And again, that's the NA of either DNA or RNA. Depends what kind of virus, depends on what it's going to make. So it's got to make the parts. It's got to make the proteins, that's going to make up the capsid, and it's going to make more genetic material. So on here, my original virus, the capsid is all kind of this lavender color, and the DNA or RNA or whatever it had is blue. So in the synthesis or replication, it's making all the parts. It's making all the capsid parts. It's making all the genetic material. Then after it makes all the parts, it assembles it, and then they get released through that lysis. So it makes the parts, then assembles it. Different pictures. I got lots of pictures. Again, we have that attachment, absorption, entry, penetration, synthesizer, replication, making all the parts. This one's red with greenish colored DNA. Assembles all of it so they look like viruses, and then gets released. This one I threw in here. You don't have it on yours, because this was the newest book with the book picture, with the new wording. Again, it's attaching absorption. So it's recognizing that very specific bacteria. It goes in reverse too. I don't like it. Penetration, it gets the genetic material into the cell. The replication, it makes all the parts. It assembles, it starts to assemble all the parts. This one adds maturation in it, usually goes together. And then lysis, it gets rid of it. Now, that's the same 5 basic steps, but some viruses can tweak those 5 basic steps. This happens more in viruses that infect humans than viruses that infect bacteria, but this can do it with bacteria. And it's known as the lysogenic life cycle. It's the lytic life cycle with a couple extra bonus steps in there. So it's a modified life cycle, so it will infect a host cell. It can infect a bacteria, but instead of the bacteria going straight into synthesis, assembly, and release, it ends. Change color. So it's still going to have that absorption. It's still going to have penetration. Then it has bonus steps right in between here before it goes into synthesis, assembly, and release. The idea is it attaches, it gets the genetic material in there, but instead of the cell becoming a virus-producing factory, that viral DNA incorporates into the bacterial DNA. And it doesn't just become a virus-producing factory at this point. It lays dormant. And as the bacteria reproduces and makes more bacterial DNA, it also starts to duplicate the viral DNA as well. Because bacteria's job in life is to duplicate. And as it makes more bacterial DNA, it also includes some of that viral DNA. When the virus is laying dormant, because the genetic material is just sitting there, not making new viruses, we call the virus is known as in its temperate phase or a prophage. It's got 2 names. It just means the virus at this point is dormant. It incorporated its DNA into the virus, but it isn't doing anything else. It's not making new viruses. It's laying dormant. It's inactive. However, when you have a bacteria, or even one of our cells, and it's got some viral DNA in it, depending what's on that virus DNA, it can change something about that cell. So it can alter the phenotype of the bacterium. Depending on where it inserted, maybe this bacteria can't make something anymore because it inserted itself right in the middle of some other gene. Maybe it starts to make something that it shouldn't have before, depending where it inserted. So depending where it inserts, it can change the bacteria. Showing an example of the lysogenic cycle and the lytic cycle. So lytic cycle, 5 basic steps. 5 basic steps. You have that attachment, absorption. you have the entry, penetration. You have the synthesis or the replication, making all the parts, assembling all the parts, and release. However, in a lysogenic, after that entry, after that penetration, instead of going straight into making more viruses, that viral DNA incorporates into the bacterial DNA. So there's your viral DNA that's going to incorporate itself into bacterial DNA. As bacteria reproduce, one bacteria becomes two bacteria becomes four, and so on. That means all of the bacteria all have viral DNA in them. Something at some point will trigger them. It's known as induction, will trigger them to go back into the lytic cycle. So they'll sit there dormant, but then something's going to trigger them, just draw around there, to go right back into the lytic cycle. Except now you don't have one hijacked bacteria producing viruses. You have potentially millions of bacteria all going back into that lytic cycle, mass producing a lot of viruses all at once. I found this little video clip. There's no sound, and it keeps repeating. Just showing the difference between a lytic cycle, cell gets hijacked, there's the viral DNA, virus-producing factory. It will go through lysis, cell dies. It will start over. In the lysogenic cycle, cell gets hijacked by a virus, viral DNA goes in, incorporates into the cell's DNA. This bacteria divides like it's supposed to, and then each new cell all will have viral DNA incorporated into it. And again, it's just waiting. Something will trigger it, whether temperature, immune system, something will trigger it to go right back into that lytic cycle. Another comparison. Lytic cycle goes in, becomes a factory, makes lots of viruses. Lysogenic goes in, incorporates itself in there, cells will reproduce with new viral DNA. Now, animal virus replication. So these are viruses that infect animal cells, and that includes us. We are in the animal kingdom. It still has the same 5 basic steps. But there are some differences that happen in these 5 basic steps because some of these viruses have envelopes and acquire them when they get released. Our cells are eukaryotic versus prokaryotic, so viruses that infect a prokaryotic cell like a bacteria behave a little different than viruses that affect eukaryotic cells like ours. And our cells don't have cell walls either. So there are some differences as we go through the same 5 basic steps. So the first step, attachment, adsorption. Again, there's going to be usually some type of chemical attraction, something our cells give off that initiates viruses to be attracted to it. And then again, it needs to use some type of glycoprotein spike on the virus to actually attach to our cells. Viruses don't just magically like, well, that looks like a cool cell. And I'm like, there has to be an attachment protein. Also, the don't have tails or tail fibers, and what that means is they don't look like the things that land on the moon. That little landing gear that looked like it was landing on the moon, that's called tails or tail fibers. Viruses that infect us don't have those. They look cool though. The entry or the penetration step can also be a little bit different. There's 3 different ways a virus can get inside of one of our cells, which are my 3 pictures. The first is known as direct penetration. And again, there's some type of protein or receptor that it likes. It attaches to it. And then it just injects, that's the direct penetration, it injects the genetic material in. So the capsid stays on the outside, the genetic material gets in. That's one way it can get in. That's one penetration, even called direct penetration. The second way it can get the virus inside the cell is known as membrane fusion. If it is an enveloped virus, so this can only happen with enveloped viruses, is if a virus has an envelope, that's a phospholipid bilayer, our cells are a phospholipid bilayer. That when it recognizes certain receptors and attaches, all of that phospholipid bilayer, all the yellow, kind of melds together. I mean, a phospholipid bilayer, it's a fluid mosaic model. It all kind of melds together. And then the whole capsid, the nucleocapsid, the capsid and the nucleic acid, that whole thing goes into the cell. So the membrane of our cell, that membrane envelope all fuse together, and then the whole nucleocapsid goes in. Again, the virus does not need the capsid inside the cell. And the first thing it will do is get rid of it. But the genetic material is still in there. The third way it can get in, envelope virus is called endocytosis. Most resembles phagocytosis. It almost looks like the cell is eating the entire virus. Virus has to recognize a receptor, it attaches, and then the whole cell membrane goes out and encloses the virus, the envelope part of the virus, the whole thing. And it brings the entire virus inside. So it almost looks like it's going up and eating the entire virus, envelope and all. It brings the entire thing in. It brings the genetic material, it brings the capsid, and it even brings the envelope. Because the virus doesn't need all of it, the first thing it's going to do is get rid of it all. It will get rid of that envelope, it will get rid of the capsid, and you're left with getting the genetic material in. So some different ways that it can get in. If you were a bacteriophage, pretty much your direct penetration and that's it. But animal cell viruses, little different, more options. We still have synthesis or replication. We still have to make the parts. We still have to make the proteins that become the capsids, and we still have to make more genetic material. There can be different strategies, though, depending if the virus has DNA or RNA, and whether it's double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA. And again, is it the positive single strand of RNA or is it the negative single-stranded RNA? The virus has to be able to make that same genetic material. Usually, DNA is made in the nucleus. Because our cells, they're eukaryotic cells, they have a nucleus, so new viral DNA gets made in the nucleus. If it's an RNA virus, that new RNA gets made out in the cytoplasm. But the different ways it can make RNA, I'm going to put all this up here. If it's making the positive single strand versus the negative single strand. They're called positive sense single strand, negative sense single strand. Again, they had to come from a copy of DNA. It's like, well, which one is which? One way to help, when it goes through normal transcription translation, you go double-stranded DNA to make single-stranded RNA, messenger RNA to make protein. Normal messenger RNA is equivalent to the positive strand. Why that makes a difference? Ribosomes can see the positive strand. And what's the job of a ribosome? Make protein. And so a ribosome can read only the positive strand. It can't read negative strands. Ribosomes can read the positive strands and read them to make proteins. So our normal messenger RNA is equivalent to a positive strand. So how does a virus that has positive single-stranded RNA, it has the positive strand, it still has to go through synthesis, it has to make proteins, and it has to make more of that positive strand of RNA. It has to make more of that. So here's our positive sense single-stranded RNA. It injects its genetic material, so now we have an infected cell. But we have to make more of this, and we have to make proteins to go through our whole replication. Well, to make more of the positive strand, it still has to bring in the correct AU, Cs, and Gs. How it does that, it first has to make a template. What can I read to then know which letters to bring in? So the first thing it will do, it will make the opposite strand. And the opposite of positive is negative. So it's first going to make the negative strand. So, for example, if your positive strand had, I'm just going to, because I said them AUCG, it's first going to make a template, writing fast, which is the opposite, and the template is going to be the negative strand. What's the opposite of A? U. What's the opposite of U? A. What's the opposite of C? G. And the opposite of G? C. It's going to make a template with these letters so that now as it's making more positive strand, it's like, oh, I see a U, let me bring in an A. Oh, I see an A, let me bring in a U. And it uses that template to bring in the correct letters. Now, what's nice, because it is still a positive strand that this virus has, and so the ribosome can read it and it can make all the proteins that it needs for the capsule, and then it assembles them all. But the only way to make more positive strand is to first make an opposite template. Now it's similar, but everything in reverse for the negative strand. So if you're a virus that has the negative strand, it does mean that the ribosome can't recognize it to make proteins. But the first thing it wants to do is make more of the negative strand. So it's going to make a template so it knows which letters to bring in. And the opposite of negative is positive. So it's going to make a template. It's going to make an opposite strand. And then it uses that to read it. And so if this had an A, this had a U, and then this is reading this and be like, oh, it had a U, let me bring in an A. And it makes lots of copies. Because the negative strand cannot get read by a ribosome, that's okay. Because when it makes its template to make more of the negative strand, the template is a positive strand. So the ribosome reads the template to make all the proteins, to make the capsid parts. And then, which is super unique and only found in viruses, there are some viruses, there's not many, there are some viruses that are double-stranded RNA. Usually RNA is single-stranded only. Now, if you're a double-stranded RNA, you have to make more double-stranded RNA, which is great. So you've got both strands, you've got both the pinky and the red, you've got the negative and the positive strand. Each one serves as a template for the opposite. So it just reads the other strands and then assembles them back together. And as long as you've got one of the strands as the positive strand, the ribosome reads it and makes all the proteins it needs. So there's a lot of genetic variety in viruses, which means our cells have to assemble them correctly to make new viruses. Viruses, as tiny as they are, have figured out how to do all this to hijack our cells. Now, we still have assembly and release. And I put them together. So DNA viruses are usually assembled in the nucleus because that's where the DNA is made. RNA viruses usually get assembled in the cytoplasm. How many viruses get made per cell usually depends on the type of virus as well as how healthy the host cell is. Some of the cells in our body don't live forever. They've got a timeline. And if you've got a cell in your body that's only going to survive for another week or two, it's not the healthiest cell. It's probably not going to be a really big virus-producing factory. If a virus is an enveloped virus, again, not all viruses have envelopes. If they are, we call them enveloped. If a virus is an enveloped virus, it steals it from its host cell during release, this is what it's showing. So the virus was already assembled, but as it was leaving the cell, it steals part of the host cell membrane. And now you have an enveloped virus. So it steals some of our own cell membrane. Now, enveloped viruses, although they're easy to kill, usually cause more persistent infections. And part of that, even though they have viral particles on the outside that our immune system can recognize, other than those little particles for our immune system to recognize, they're coated in our own cell membrane, which is a really great camouflage. I mean, our immune system can recognize these proteins, but it may take them a little bit longer. So it can cause longer lasting infections till your immune system gets rid of them. You also notice when this virus is leaving the cell, Is it destroying the cell when it's leaving? Nope. And so again, our cells become virus-producing factories, and the factory doesn't explode, which is better for the virus. They don't all cause lysis of the cell. So naked viruses can get released by cell lysis and bust apart, again, breaking the cell. But they can also get released but through exocytosis, which would look similar to that viruses can just leave the cell without killing the cell. Eventually, they start to kill the cell. Now, there are some animal viruses, again, these are viruses that infect animal cells, we're animals, that again, don't cause our cells to right away start making lots and lots of viruses. They can go through what's similar to that lysogeny. The virus genetic material incorporates into the host cell, and then it hangs out and remains dormant. They're known as latent viruses, also known as proviruses, and they hang out in the cells for years, if not your entire lifetime. Again, that viral genetic material is inside of the cell. Might not be doing much, but the genetic material has incorporated into our own genetic material and it's hanging out. Couple examples. HIV incorporates itself into our genetic material. Even though we have lots of drugs to try to keep it in that dormant phase and not go back to making new viruses and through that, once that virus is in the cell, it's there forever. Like you will always have HIV. You might not be making new viruses and shedding those viruses, but you will always have HIV. Same thing. Herpes, we're going to talk lots about herpes on Wednesday. So much herpes. The whole herpes family of viruses, almost all of them incorporate into our DNA and then stay with us for very long periods of time, depending on which herpes, because there's 8 different herpes viruses that affect us. Depending on which herpes, some last longer in our bodies than others. It's because they incorporate. And then again, things can cause them to go active and go through this, but otherwise they're with you. Just a side-by-side comparison. Looking at those different phases for bacteriophages, viruses that infect bacteria versus animal viruses. Again, same 5 basic steps with some slight differences. Now, how do viruses cause cancer? I'm going to draw some stuff up here shortly. Now, a couple notes about what is cancer and what causes cancer. So cancer ultimately is uncontrolled cell growth. Cells just keep growing and nothing ever tells them to stop. And when cells just keep growing and growing and growing and nothing ever tells them to stop, usually what happens is you start to get mutations and nothing ever tells them to stop. Nothing ever tells them to stop, and so cells start to keep growing and they're not doing what they're supposed to be doing. Now, in our bodies, to try to prevent cancer, our cells do have checkpoints to try to stop cancer from ever happening. So we've got genes on our cells that can stop cell division altogether. And we can activate those genes and say, okay, well, there's something going on, we just need to stop cell division. Ultimately, stopping those cells in their track. We've got genes that can prevent unlimited division, which means we've got genes that will allow cells to reproduce and replicate, but only for a short time. We've got genes that control just cell division overall that we can turn on, and we have other genes that inhibit cell division that we can turn off, and then we've got genes that inhibit or stop division that we can turn on. We have a lot of genes. Again, genes are snippets of DNA that control something. We have a lot of genes to stop, turn on cell division or turn on inhibitors for cell division, and we've got genes we can turn off for cell division. Again, if cells are not replicating correctly, we've got genes, hopefully, that will stop those cells from dividing. But if that doesn't work, I'm going to draw stuff while I'm talking. If that doesn't work, you end up getting what's known as a neoplasia. What colors? Hold on. Purple. Which just means new growth. Anything neo means new, like neonatal, means new. And plasia means growth. Now, if you have cells that are growing uncontrolled, you're going to develop a tumor. Now, there are two kinds of tumors, benign tumors and malignant tumors. If you had to have a tumor, which one would you want? A benign, because that means the cells were growing, growing, growing. Nothing told them to stop for a while. And they stay put. And so they remain in place. So those cells were growing uncontrolled. They're abnormal looking, but they stay put. Malignant means that cells were growing, growing, growing. Now they're breaking off. They're getting into bloodstreams, lymphatic system. They can end up anywhere. And then wherever they end up, they still are growing uncontrolled. That's what you don't want. Now, this is what I'm trying to draw with colors. So just ignore me for a minute. Glad I had lots of colors now. I'm like one color short. I'm going to explain all of this in a second and define these. All right, a few terms. Oh, gene. Now, when we're talking cancer, again, cell division. And when we talk cancer, cell division is bad because we're talking uncontrolled cell division. What does onco mean? You're in the oncology unit. Means cancer. Now, this is a gene. Again, it's a snippet of DNA that controls something. And an oncogene means this is a gene that causes cells to divide, aka cancer. When we're talking cell division, we're talking that's a bad thing. That's uncontrolled cell division. So an oncogene, this is a cancer-causing gene. And why it causes cancer is because it's a gene that turns on cell division. Now, when we stick the word proto at the beginning of something, proto, just like if you stick like a prologue to something, means like before, which means it hasn't actually happened yet. So a proto-oncogene is inactive. It actually isn't, it's not doing this yet. It's like a prologue, the book hasn't happened yet. It's just some background stuff. A proto-oncogene, it hasn't started cell division. So I like to use smiley faces and stuff. Sad faces for cell division, smiley faces for not cell division. So a proto-oncogene, is this going to cause the cells to divide? If it's in the proto-oncogene? No. It's not doing anything yet. Nothing's telling the cells to divide. Again, it's got the information, but it's not active yet, so we call it a proto-oncogene. We also have repressor genes, which means they repress or stop cells from dividing. If the repressor gene is here and it's present and nothing's interfered with it, will cells divide? No, because it's repressing cell division. So we're good. no cancer. But what can happen is if you get a virus, and it's a virus because not all viruses can do this, but if you get a virus infection and the viral DNA incorporates into our DNA, because these are snippets of our DNA. Let's say it incorporated right there. You've got some viral DNA that inserted itself right before that proto-oncogene. And because it inserted right there, it activated this. Now, will an oncogene, not proto-oncogene, cause the cell to divide? Yep, and in our case, that's a bad thing. However, we still have a repressor gene right here that's going to stop cells from dividing. So at this point, we're still no cancer. It's generally not the first, they call it the first hit. It's not the first virus that causes issues because we've got lots of genes that are trying to stop this from happening. But if you had viral DNA that incorporated itself right here, we already said that's bad because now you've got this active oncogene that's trying to make your cells to divide uncontrolled. But if you get more viral DNA, say from a different virus or even from the same virus, that incorporates itself right smack dab in the middle of the repressor gene. If you interrupt a gene, can it do what it's supposed to do? No. Which means this repressor gene can't stop cells from dividing. This is now when you end up with cancer. So viruses are known to cause a lot of different types of cancers. Not all cancers are caused from viruses, but a lot of them are. And it's because the viral DNA gets inside of our cells, and if it incorporates into our DNA, depending on where it incorporates, does it incorporate right next to an oncogene? Does it incorporate right next to one of those repressor genes? It can lead to cancer. So again, there's lots of other things that can cause cancer: UV light, radiation, chemicals, but viruses are one cancer-causing thing out there. Viruses cause about 20 to 25% of all human cancers. That's a lot. I mean, not all. There's different genetics. Again, genetics, we're talking your genes are messed up somewhere in these repressor genes. But there's a lot of cancers that are caused by viruses. Couple examples: Burkitt's lymphoma is caused by the Epstein-Barr virus. Hodgkin's disease is caused by the Epstein-Barr virus. Kaposi's sarcoma is caused by HIV. Cervical cancer caused by HPV, although it can cause other cancers as well. Now, we're very fortunate these days. It's because we have a vaccine for HPV. It's like our first vaccine that can stop cancer. If we can stop the virus, we can stop some of these cancers from happening. It'd be great if we had vaccines against Epstein-Barr and if we had vaccines against HIV, maybe. Somewhere in our lifetime, hopefully. Again, potentially, if we can get vaccinated against all of these viruses that cause cancer, we'll eliminate 20 to 25% of all cancers. Awesome. Now, if we wanted to grow a virus, there's a few different ways. Again, to grow a virus, you first have to grow the cell that it needs to get in, and then you can grow the virus. So different things that we can use to grow viruses. If it's a bacteriophage, we first grow the bacteria that it infects, then we can grow the virus. If it infects plants, we first grow the plant. Again, if it infects certain animals, you gotta have the animals. So depending on the organism, or depending on the virus, might depend on which organism you have to use. Another way we can grow viruses is we use embryonated chicken eggs. Again, it's one cell. It's a really big cell, which means we can inject it with a virus and it will take hold of that cell and it becomes a huge virus-producing factory. And it is one of the reasons why, again, some vaccines are made this way, is we can grow the virus, we can alter the virus so that it can't cause disease, but it can wake up our immune system. Otherwise, we can grow cells in the lab. It's usually in a nice nutrient pink fluid in the lab. So we can grow cells in the lab, then we can infect those cells with viruses to grow the viruses. Different reasons why we want to grow viruses, other than vaccines. Sometimes we want to grow them for research. We want to research the virus. We want to research its replication. How is it hijacking cells? How is it reproducing? Is it doing through lysis? Sometimes we just want to see, what fully does it do to the cells? Maybe it doesn't even really affect the cells all that much. Or does it fully destroy them through lysis? Some we can use it to actually diagnose viral infections. If I go back to way back to the immune system, if we can grow cells, and then we can look at a patient's cells, if they had antibodies to a certain virus, the cells in the lab wouldn't die. If the patient didn't have antibodies and we mix it with viruses and with cells, the cells would die. So sometimes we grow viruses to diagnose patients. There's lots of reasons why we want to grow them. A lot of times it's research. The question, are viruses alive? There's two competing ideas that they are complex pathogenic chemicals. So no, they're not alive because they don't meet the basic characteristics of life. They can't live on their own. They're not a cell. Other people say, but how can we not think of them as alive? And so let's call them alive, but we'll just call them the least complex living entities out there. Because they are pretty complex. For not being a cell, all they are is a capsid and some nucleic acid. They hijack host cells. They completely manipulate them. And they can cause a lot of issues. So some people call them just the least complex living entities out there. I'm one of those, I don't like to think of them as alive because all they are really is a protein and nucleic acid, but it's that nucleic acid that causes so many issues. Now, my last little group, going away from viruses, are prions. Prions are not viruses. We talk about them in this unit, though, because they are also not living entities. They're not cells. They're just a protein. So they don't even have nucleic acid. It's just a protein. But it's an infectious protein, which means it's a disease-causing protein. So we talk about them with viruses because they are so simplified. A virus is just protein and nucleic acid. Well, this is just protein. So a prion gets its name because it's an infectious protein. So it's a protein that's infectious, and so they called it a prion. It's a very specific protein. It's called, it's a change to what's known as a protein P. That's what the PRP for. A normal protein P, this is the structure. It has these, they're called alpha helices. They've got the little twist, the old telephone cords. That's the normal structure of this protein. Now, from what you know of proteins, if you change the shape of a protein, can it do its job? No, you've denatured the protein. A changed protein can't do its job. This is the normal structure of this protein. An abnormal protein P looks like this. It's called a prion, PRP. Instead of having these little helical shapes, they're called beta sheets. They're like straight sheets. But just changing a protein, you're like, normally your cell would be like, well, that's abnormal, let's get rid of it. And it doesn't cause any further issues. Unfortunately, with a prion protein, it doesn't do that. The problem with prions is this abnormal protein causes other proteins to change shape. That's what makes it infectious. This changed protein, instead of just being abnormal and not doing what it's supposed to do, causes other proteins to change shape. So it tells all the neighboring proteins that look like this, like, hey, it's not cool anymore to have those helical shapes. You got to have straight shapes now. And then these proteins are like, oh, okay. And then they untwist and turn into this. And more of these proteins, these prions, cause even more of these proteins to change shape. That's why it's called infectious, is because it starts to spread. Abnormal proteins cause other normal proteins to change shape and become abnormal. And one of the biggest issues is that this particular protein is found in our nervous system. And so prions affect our central nervous system. They affect our neurons. They affect our nerves. They cause, there's not a lot of prions that cause disease, but the ones that do are catastrophic because it causes your proteins in your nervous system to change shape, which means they can't do their job. And the problem with it, it's a slow progression. This is not like a virus or a bacteria where you start to see signs and symptoms within days or weeks, years, if not decades. That it's a very slow degenerative progression. Mm-hmm. Yep. Yep.
I had someone at the hospital who passed away from that, and like she went from a month ago watching her kids to full on like couldn't speak or nothing.
Yeah, and unfortunately, there's nothing. We have no treatment. Like the damage is done. So there are different prions. Again, because it affects their central nervous system, it affects their brain tissues. It ultimately causes a condition known as spongiform encephalopathy, which I was like, oh, that's a really fancy, weird, horrible disease name. The word pathy means disease, encephalo means brain. So it causes a condition where your brain looks like a sponge. Is your brain tissue is breaking down because that protein can no longer do its job. And so your brain tissue starts to have all these little open cavities, aka starts to look like a sponge. And you can't go back in and fix that once the damage is done. So it's a fatal neurological degeneration. It's a very slow progression. Well, I shouldn't say that. Some are faster than others. Others could be decades later. We pick up these prions. These are not things we're born with. Occasionally, you can have a protein that goes rogue and changes into a prion. That's rare, that it will just all of a sudden spontaneously become a prion. We normally pick up these infectious proteins by ingesting contaminated meat or contact with mucous membranes. Again, it's rare. A couple of examples, the BSE, I was going to say, these are going to be on my next slide. The BSE is bovine spongiform encephalopathy. Any guess what that caused? Or what animal a bovine is? It's a cow. Now, cows could get these too, and it causes their brain tissue to look like that. What's the nickname for the disease? Mad cow disease, because is the cow going to act normal if its brain looks like this? No. Hence the mad cow disease. And then we could pick it up by eating infected beef from those cows. The VCJD variant Creutzfeldt-Jakob disease and CWD, where have you heard that? You hear it in the fall. Deer. It's found in deer. So this was showing difference, spongiform encephalopathy, normal brain tissue. These are animal prion diseases. Again, the problem is if we eat infected tissue, we found out that we could get this from eating infected beef from animals that were infected. Otherwise, human prion diseases, Creutzfeldt-Jakob disease, I don't even want to try to say that one, fatal familiar insomnia. Kuru's gone away. This was only found in a certain African tribe. And they realized, why is everyone getting this one very random prion? And then they realized in that African tribe, it was customary to eat the brain of the person that died to keep to keep them alive forever. Well, that's the transmission sequence. So they stopped that and it went away. But it is around. The biggest question that we don't know the answer to yet is we know that humans were able to pick up this prion from eating infected cows. So what happens if we eat infected deer? Can we get it? We don't know. We don't know. So they're pushing you to test your deer during hunting season. And then if your deer tests positive for this, well, it's on you now. Do you want to eat it and take that risk? 'Cause we don't know the answer. As of right now, we don't have any documented cases of we know someone ate infected deer, now they have a prion disease, but it could be 10, 20, 30 years from now before we even would notice. I don't think anyone's going to be like, hey, last year I ate a deer, was acting kind of weird. Could you take some brain sample tissue and test me? No one's generally going out there and doing that. So a lot of times we're not going to know the repercussions or if that's a possibility. I don't know. We have our deer tested. None of them have tested positive yet. I don't know what we're going to do.
My uncle's last year tested positive.
Did they eat it or did they? No. Yeah, it's one of those I'm like, I love venison. But it's one of those I'm like, I don't know. We don't know the answer yet. And I expect somewhere in our lifetime we'll find out. But even still, if someone ends up with one of these diseases, could you know for certain? Was it, because we haven't tested deer until the last few years. Like, did you eat an infected deer? Did you not eat an infected deer? How do we know 20 years ago what caused, your current infection? So this was showing them like CWD is found in Wisconsin. I couldn't find a more updated 2023 results from last year. I know they've got it out there, but not a cool map. So lots of testing is taking place. That's what the darker red is testing. The numbers are how many number of deer actually have tested positive. So we are doing lots of testing. It seems more testing is happening more south in the northern states, but I'm like, we do have deer that test positive for CWD. Just showing. There's lots of, sample test sites you can drop stuff off now. So we're going to end there with all that happy news.